Trinity Cultural Academy, Coimbatore and New India Publishing Agency, NEPUS, New Delhi welcomes you all for this presentation on hybrid seed production in maize, tidbits 20. And this content comes under the major subject plant science and seed science and technology. And this material is highly useful for ICAR All India Entrance Examination, JRF and SRF for PG admissions, ICAR NET, ARS and STO examination. Apart from this, it is also highly useful for IBPS Agriculture Field Officer examinations. And this material is prepared by Dr. K. Vanangamudi. He is the formerly Dean Agriculture, TNAU Coimbatore, and Dean Adiparashakti Agricultural College, Kalavai, and Professor and Head Seed Science and Technology, TNAU Coimbatore. And in the previous video, we have seen the hybrid varietal seed production aspects in maize, and in that the biological characters are also seen. Here we are going to see hybrid seed production in maize. The main important tool employed for seed production in hybrid maize is detasseling, and it is a manual emasculation by detasseling, and detasseling is nothing but a physical removal of male part of inflorescence, male part from the female plants by allowing the plant to act as a female, which in turn crossed with the selected male plant and affect seed setting. This is possible in maize alone because the crop is monoecious in nature and also protandry nature of flowers. Monoecious is nothing but the plant which is having both male and female flowers in the same plant but at different position, and the protandry nature is nothing but the androecium, that is the male part of the plant, matures first. And the detasseling, already we have seen the tool employed in hybrid seed production in maize is detasseling. Tassel is the male inflorescence of maize. Detasseling is nothing but a removal of tassel or male flower from the female plant. Detasseling should be done in the female plant of hybrid alone, and it should be removed before anthesis and immediately after emergence. And the detasseling should be completed in well out of the boot leaf but before the anthers shed the pollen. It is done every day. It is done in every day from the emergence, that is from the anthesis up to 14 days. And the anthers day is 2 to 4 days to days after complete emergence. And it should be, the tassel should be emerged out of the boot leaf, should be removed, but before anthesis they have shed pollen. And the method, the procedure of detasseling is, the stem is to be held with the left hand and the tassel is to be removed with the right hand in one upward pull. And grasp, the pulled tassel should be taken away from the field and buried beyond the isolation distance. In any case, no spikelet should be left, which may cause genetic contamination, and the leaves also should not be removed as it favors reduction in seed yield. Here the tassel should be removed, the entire tassel should be removed so that all the pollen parts are fully removed. So, and also do not break or remove leaves, as removal which reduces yield and ultimately leads in lower quantity of seed. This is how the detasseling technique takes place, by holding the stem in left hand and pull out of the tassel in right hand in single pull. And the important precautions to be taken while detasseling is, do not Don't allow improper detasseling, immature detasseling or incomplete detasseling. Improper detasseling is nothing but a portion of tassel is remaining in the plant while detasseling. Or immature means carrying out detasseling work when the tassel is within the leaves. Or incomplete means tassel is remaining in lower or unseen or unaccounted place in within the whole of the leaves. There should not be any shedding tassel. In case the shedding tassel is present in the plant, it may contaminate the genetic purity of the crop. It is not shedding tassel is nothing but either full or part of the tassel in the female line is nothing but a shedding tassel. And this is the source of contamination of genetic purity in hybrid seed production. The system of hybrid seed production in maize is nothing but a detasseling, that is the manual creation of male sterility in female line. And there are different types of hybrid in maize. Single, double and three-way cross hybrids are available. Let us see one by one in detail. Single cross hybrid is the first one is single cross hybrid. It is the cross between two inbred lines where one serves as a female and the other one serves as a male. Inbred A line serves as a seed parent and it should be detasseled, that is the female line, and inbred B line act as a male parent, that is the pollen parent. Here A and B are crossed. Two inbreds are crossed to obtain a single cross hybrid. The A genotype will be detasseled and crossed with the B genotype. The hybrids obtained from single cross hybrid are CoH1, CoH2 and CoH5. The parental lines are given here. Kindly go through this. And the double cross hybrids are nothing but it is a cross between two single cross inbreds which involves four inbred line. Cross between two hybrids, A cross B, which is crossed with the C and D. A and B, A cross B single cross hybrid will be produced by detasseling A and by crossing with B line, and C and D hybrid will be obtained by producing obtained or produced by detasseling C line and crossed with the D line. And then A cross B is crossed, will be detasseled and crossed with the C cross D hybrid. It acts as a female parent and this one acts as a male parent. Double cross hybrid examples are Ganga 2, Ganga 101 and CoH3. Kindly go through the male parental lines here. And in case of three-way cross hybrid, cross between a single cross and which serves as a female and crossed with an inbred line which serves as a male parent. First generation resulting from crossing approved inbred line and certified open pollinated variety with the inbred variety. A will be detasseled and allowed for crossing in the variety. Ganga 5 and CoH4. These are the examples of three-way cross hybrid. And the last one is double top cross hybrid. This is nothing but a controlled crossing of certified single cross and a certified open pollinated variety. This is how the crossing takes place, and A cross B hybrid will be detasseled and crossed with the open pollinated variety. Let us see the seed production technologies here. The season followed for hybrid seed production is nothing but during November, December. The pollination should not, the best season is November, December. The other seasons July, August, January, February or September, October. And the isolation distance followed here is in case of inbred line or varieties or synthetics composites, it is 400-200. In case of hybrid seed production, here they have given isolation distance of foundation seed here. Here 200. And for field standards for isolation, which is modified based on the situation, or and in case of same kernel color, the foundation seed isolation distance is 400, 200. Different kernel color means 600, 300. Field of single cross inbreds not conforming to varietal purity means 400, 200. Single cross with same male parent means 5, 5. It is enough for single cross with the same male parent. In case the single cross with another male parent means 400, 200. Mostly 400, 200 is followed for hybrid seed production of maize, 400, 200. And in case of hybrid seed production, this we have already seen in varietal seed production, that in hybrid seed production, particularly certified seed production stage alone, the isolation distance which is less than 200 meter can be modified by increasing the border rows of the male parent. 2 to 4 border rows are maintained in hybrid seed production. This can be increased by decreasing the isolation distance. If the kernel color and texture of the contaminant are same as that of the seed parent, if the kernel color and texture of the contaminant are same. It is determined based on the size of the field and its distance from the contaminant and shown in the table. Kindly go through this table. If the area is less than 4 hectare means the isolation distance 200 means the number of border rows is 1. In case it is greater than 16 hectare, the isolation distance is 15 meter, the border rows is 13. So kindly go through this table for the adjustment which are taking in hybrid seed production for border rows. And the spacing here is, since it is not a transplanted crop, it is not sown in nursery stage. The seeds are sown directly in the field in ridges and furrows. For hybrids, the spacing followed is 60 by 25. Particularly, female line is 60 by 20 or 75 by 30. For male line, 45 by 30 centimeter. For seed rate, the female line has 7 to 10 kg per hectare and the male line has 3 to 4 kg per hectare. The planting ratio, that is between female and male line ratio is, in case of single cross hybrid, 4:2 ratio, 4 lines of female line and 2 rows of male line. In case of double cross, 6:2 and 3-way cross also 6:2. This is how these 2 lines are male lines and this 4 lines are female lines, 4:2 or 6:2. And in case of border rows, inbreds and single crosses need 4 rows of border rows. For other double cross or 3-way cross like that, 3 rows of border rows is enough, and it may vary depending upon the conditions as shown here. And next one is fertilizer. The NPK requirement, fertilizer recommendation for hybrid seed production is 200, 100, 100. It is applied in 3 split doses, one as basal 100, 150, and in first top dressing on 20th day after sowing, which is at a vegetative stage, 50 kg of nitrogen is applied, and in second top dressing 50 kg of nitrogen and 50 kg of potassium is applied during boot leaf stage at 45 days after sowing. In foliar spray of 2% DAP at 50% flowering stage is applied to enhance the flowering. Ultimately, it will increase the yield of the particular crop. And in zinc deficient soil, zinc sulfate at 25 kg per hectare is applied. And the most important operation for hybrid seed production is roguing. Here, shedding tassel are removed from the field. Shedding tassel is nothing but some of the tassel which may remain inside the boot leaf during detasseling due to improper removal of tassel. This may shed pollen and cause genetic contamination. Hence, detasseling should be perfect without shedding tassel. And apart from the shedding tassel, Roging, it's nothing but a removal of unwanted, off-type and diseased plant from the seed production plot, and it is done based on leaf variness, tassel color, cob shape, stem color, silk color, number of leaf and presence or absence of auricle. And the weed management aspects and irrigation, these are all the same as that of varietal seed production. Let us move on to the field standards followed here. We have already seen that the isolation distance is mostly 400, 200 and it may vary depending upon the specific character. And in case of off type, for foundation seed it is 0.2 and for CS 0.5, certified seed 0.5, shedding tassel 0.5 and certified seed is 1% is allowed. And inseparable other crop objectionable weight designated should be nil. And specific field standards are off type shedding pollen when 5% or more of the seed parent in receptive cycle 0.5. And kindly go through this table. This is all more important in examination point of view. Kindly go through this. And the next important one is seed inspection. Seed certification officers carry out the seed inspection in 4 stages of hybrid seed production of maize, 1 before flowering and 3 during flowering, to manage or to improve the quality of the seed production in hybrid maize. And the next one is harvesting. Harvest when the moisture content falls below 20 to 25%. Harvest male line first and remove from the field and then harvest female. The physiological maturity stage is when the stalk color changes to yellow color. This is how the physiological maturity, or this is a maturity stage where harvesting takes place. And the next important operation is threshing. Here there are 2 operations as dehusking and cob sorting, and xenia, metaxenia, these operations and all we have already seen in varietal seed production. Kindly go through those materials, and here let us see at a glance. Dehusking is nothing but husk are removed manually, and cob sorting is nothing but removal of ill-filled, diseased or cobs having kernel color variation or separated and packed. Xenia is nothing but effect of pollen on endosperm and related tissues in formation of seed, that is, which leads to seed color. In maize, gene present in sperm cell contributes the expression of color of hybrid seeds. Metaxenia is nothing but effect of pollen on the maternal tissues of the fruit. Shelling is nothing but removal of maize kernels from the shank. Cob sorting should be the first operation as a post-harvest evaluation for genetic purity, and the sheath is removed and checked for kernel color, shank color and diseased cobs and etc. And at the final stage, shelling takes place. Cobs are shelled either mechanically or manually at 15 to 18% moisture content only. Improper shelling leads to 48% damage to kernel. So proper shelling operation should be carried out as it may damage the seed quality. And crack on pericarp can be identified by ferric chloride test or tetrasodium chloride test. Shelling is done mechanically using cob sheller. Nowadays for a large scale production, cob sheller is used, and manually by rubbing with the stones. Then the seeds should be dried to a moisture content of 12% and allowed to seed treatment of slurry. Treat the seeds with 8% moisture content, either Captain or Thiram, for storage of seeds, and the treated seeds can be stored for 1 year in cloth bag. Others as in varietal seed production like post-harvest, mid-storage corrections and pest, disease, etc. And the seed yield is ultimately 2.5 to 3.6 tons per hectare. And the important aspect in examination point of view is seed production, seed standards. Field standards already we have seen. There are 4 inspections followed for hybrid seed production, 2 inspections for varietal seed production. And in case of varietal seed production and hybrid seed production vary according to the isolation distance vary according to the kernel color based field, etc. And in hybrid seed production 400 is to 200 is followed. For foundation seed 400 meter and for certified seed 200 meter. The physical purity minimum of physical purity percentage in seeds as in case of seed standard is 98%, and the germination percentage is 90% for hybrids of maize, and for moisture content for moisture vapor pervious container is 12%. Per moisture proof container means maximum moisture content is 8% for storage, for long term storage. Thank you friends. Thank you for watching. Kindly subscribe our YouTube channel to get the upcoming videos. Thank you.